All right, keep it clean. We are recording. All right, so we left off last week kind of real quick. We were talking about this antigenic shift, right? Antigenic shift and antigenic drift in viruses. And we were talking about how you would have antibodies. So here we have antibodies, so you got vaccinated, so you have antibodies against all those antigens, so your flu virus cannot gain entry into the cell. It's neutralized, no infection. But then next year, the flu vaccine you got gave you the black and pink antibodies. The black antibodies still work, but your pink ones are no longer functional. So now you have a higher risk of that flu virus still being able to attach. So in this one, we see disease. Now, in antigenic drift, this happens in the flu virus almost on a yearly basis, and it happens quite often. And this is why we have to come up with a new flu vaccine every year. And this is also why that flu vaccine is the result of a prediction of this new mix. Notice it has this blue gene here that it didn't have in the other one. So that's why it's a blue antigen, and we have no blue antibody. Now, in antigenic shift, what we get is a completely new virus. You see what we had here with H1N1. They call it swine influenza. And so H1N1 was a flu virus that was known in pigs for over 50 years. It had been around forever in pigs, but it had been followed because all pigs are big mixing vessels. So all pig viruses are always very closely followed by the World Health Organization and the CDC, because they are there. It is known that the pig physiology allows for mixing of multiple species of flu viruses in particular. So we had a human flu virus and a pig flu virus infected the same cell. And when this occurs, we had a total recombination of the genetics of the two, and this resulted in a new virus in which none of our existing antibodies, no matter how many flu vaccines or flu diseases you actually survived, we had no antibodies against it, because this was a completely new, novel virus. Novel means new. So we had absolutely no resistance to it. And that, of course, is what's going on with the COVID virus right now, is we've never seen this virus before. It's never occurred in the human race before. So because of that, we have no existing immunity. Doesn't mean that we can't develop immunity, but we don't have any existing immunity. This isn't something that we have a vaccine against, or that we have a whole, people throughout time at a much slower rate have gotten the disease and survived. We don't have what's called passive resistance in infants, because mothers have never had it. And so mothers oftentimes pass resistance to diseases onto their offspring for protection, for short-term protection. So all of these processes that our immune system and that in biotechnology we have come up with to create some kind of immunity to a disease, none of that exists against COVID. So this COVID virus is a novel virus, and it's moving through. It has a really high transmissibility rate. It's a very scary virus. So there's a lot of concern about this transmissibility and the death rates and overcoming the healthcare system and all of those sorts of things. We will start seeing declines in cases as we start seeing more and more survivors. When people start surviving, they'll have that instilled immunity. They're going to have immunological memory. All those things we learned about in the immune system and memory, those will start coming up, and those people will be immune. If they're immune, then that's one reason they're talking about the resurgence in fall. So as people start to come back out, they start lifting this quarantine, and people start coming out and holding events again. There are going to be people who either survived it and still carry it, or people who are asymptomatic. The virus will still be within the population, and they will transmit it again. So once everybody comes out of quarantine, we'll probably see a spike in cases again in the fall. However, it will not be the same level that we're seeing today. And that is because we will have people who have existing resistance, and those people will be immune to the disease. So what they're hoping to do is, as we create more and more survivors, we can start getting just low levels of herd immunity. And as that herd immunity builds up, we see fewer and fewer cases. But there will still be some transmission even among those that are survivors of it. So let's talk a little bit about some of the different things that bacteria do and the different types of toxins that they produce that instill these disease states. We have in toxins produced by bacteria, we have endotoxins. Endotoxins are basically the LPS, that lipopolysaccharide, plus the toxin A that it's attached to, that's found in that outer lipid layer of gram-negative organisms. So endotoxins are specific to gram-negative organisms. Now, all gram-negatives with that outer lipid layer have that LPS attached to lipid A or toxin A. There may be some genetic variances of this LPS and the toxin, but they all are basically the same. They are anaphylatoxins, so they bring out strong inflammatory type signals. They also oftentimes instill fever. Exotoxins are produced by both gram-positives and gram-negative organisms. These are usually some type of either metabolic waste product or some kind of enzyme released by bacteria to break down really big macromolecules they find in the environment so they can absorb or take in the monomer units of those macromolecules. Sometimes these are just metabolic waste products, or they may be compounds that the bacteria are excreting in order to evoke another reaction. There's always something going on with these toxins that they're trying to do. So there are three primary classes of toxins. We have superantigens, AB toxins, and membrane damaging toxins. Now, endotoxins, again, are that LPS with toxin A, and these toxins are released from the outer membrane. One issue with them is they are heat stable. So even if the patient runs a fever, fever is oftentimes used by the immune system to try and reduce microbial growth, either to destroy bacteria or slow down the growth of bacteria organisms. That's one of the purposes of fever. And so these guys will induce fever. Our cells respond by inducing fever, but the toxins themselves are heat stable. So even if the bacteria don't die, or even if the bacteria do die, when they die, they still release that toxin, and the toxin is viable in a higher temperature. If released in high concentrations, can cause a patient to enter into what's referred to as septic shock. This occurs because these bacteria, this is usually occurring in a patient who has a septic infection. They have a sepsis, and that is bacteria in the blood. So if there's a bacterial infection within the bloodstream itself, we have a high concentration of these bacteria growing in the bloodstream, in the circulatory system. Oftentimes, if the bacteria all die at once or die in very large numbers, say either through fever or even through injection of antimicrobial compounds or drugs, if we kill too many at once, all of those gram-negative bacteria are going to release their LPS. That LPS is already there. So once they start dying off, that outer membrane starts to fall apart. The lipid A toxin is then released into the bloodstream in a very high concentration. This will cause system-wide inflammation, which causes a massive drop in blood pressure, and that's what shock is, right? So if you think about it, in the inflammatory response, we have that vasodilation. And if you have vasodilation occurring system-wide throughout the entire circulatory system, you're going to see a massive drop in blood pressure, and that's kind of the definition of shock. If the infection is a local infection, a non-sepsis infection, then you don't have the same kind of activity. It's not considered as dangerous. Oftentimes, if you're in the hospital and you have somebody who has a gram-negative septic infection, when they start giving antibiotics, the patient will oftentimes get worse for about 12 to 24 hours before they get better. So there are often sometimes antitoxins or other medications they may give prior to or in combination with the antibiotic in order to avoid a septic shock type of situation. In gram-positive organisms, you don't have that issue because gram-positive organisms do not contain toxin A. Now, another problem we can have is our own immune responses can cause this endotoxin to be released as well. So the toll-like receptors on some of our cells actually detect that lipopolysaccharide, and that's going to, of course, increase opsonization and antigen presentation, and thereby increase the inflammatory response. So in the exotoxins, now remember, exotoxins are produced by both gram-positive and gram-negative. They're found more often in gram-positives, but we do see them in gram-negatives as well. And these are proteins, and proteins get secreted by the bacteria. Most of the time they're proteins. Sometimes there are some lipid ones and some carbohydrate ones, but for the most part, they're some kind of protein. When that protein is excreted, it has some kind of function that's going to benefit the bacteria, right? So each exotoxin has its own really kind of unique function. Now, the first are the AB toxins. An AB toxin is a what we call this is a quaternary, I had to think about it for a second. This is a quaternary protein. It's a 2-polypeptide chain protein. There's 2 parts to it. So there's an A subunit and a B subunit, hence the name AB toxin. The A subunit is where the effect of this toxin, right? This is the effector portion. So this is where the that protein is going to make something happen inside of a cell. The B subunit is what actually binds the toxin to the cell. And 2 examples of an AB toxin are diphtheria and cholera. So here we have a receptor on a host cell. This is the outside of the cell out here, and this, of course, is the inside, the yellow is. And we have a receptor on the cell that bacteria have produced an exotoxin that is specific for that receptor. So this guy's going to bind, and when he binds, the B portion will stay bound. The cell's going to take this in, and then the A component will enter into the cell and carry out some sort of function. Many AB toxins act on what's called GPCR receptors. These are a class of receptors found in our cells that make all kinds of different things happen. But we have a high concentration of these types of receptors in the gastrointestinal tract and in the respiratory tract. So cholera AB toxins and diphtheria AB toxins are both quite dangerous toxins because of that. I'll show you guys some videos on these too. I have to pull them up. I had them the other day, but we didn't get this far. So excuse me, I'll pull them up for you in a minute. Then we have the super antigens. The super antigens are toxins that are actually very, very dangerous. We see them in what's called TSS, or toxic shock syndrome. They're produced by a couple of different bacteria. In particular, Streptococcus produces them and Staph aureus produces them, or staph species in general. And what happens here is on the left, right here, I have what would be normal antigen presentation. Remember from the adaptive immune lecture that our cells our macrophages and dendritic cells, when they present an antigen, they present it to a T cell, and that antigen is presented on an MHC class 2, that antigen presenting complex. And that antigen gets trapped between, and when an antigen presenting cell presents it to the T cell, everything is good. Everything works, it fits. The T cell says, okay, I recognize that antigen. I'm going to go see if B cells do the same thing. Now with a super antigen, what's really happening, this picture's a little deceiving, is instead of binding and connecting the MHC to the T cell receptor, it actually binds outside of the two. It actually binds out here. And it brings these 2 receptors smack up against each other, and we get an overstimulation of T cell proliferation. So it tricks the T cell into recognizing it, and the T cell, by binding it so tightly, the T cell over-responds. And we get way too many T cells produced, and they produce way too many stimulatory molecules, and we get kind of this like crazy cytokine type of reaction. It's sometimes referred to, yes, it's a cytokine storm. That's exactly it, Megan. So when this happens on a higher level, as these T cells begin proliferating and becoming overstimulated, your patient can go into what's called a cytokine storm. And a cytokine storm is kind of like your immune system throwing, yes, that's what they're talking about with COVID, Megan. In a cytokine storm, the immune system is really throwing everything it has against the wall to see what sticks. I don't know how else to describe it. Your immune system is just going, I am so overwhelmed now that I'm just going to produce everything. And it produces every possible cytokine, dumps it into the bloodstream with the hopes that something will work. Cytokine storms are an immune system's last-ditch effort. So if a patient enters into a cytokine storm type situation, it is very grim. It's not often that people survive that. It's the immune system really just kind of going haywire, like it has been short-circuited. The last type is the membrane damaging toxin, and it does exactly what it sounds like it does. It starts to disrupt or cause some kind of damage to the actual plasma membrane itself. So think about the hemolytic activity. Streptolysins are also a class of hemolysins produced by strep. The streptolysins are what we observed in the blood agar plates in lab. And hemolysins are produced by all kinds of different bacteria. Streptolysins are specific to the strep species. So think about toxic shock syndrome for a second. In TSS, we have either a strep or a staph sepsis. Oops. There we go. So we have either streptococcus or staphylococcus growing in the bloodstream, in the circulatory system. If something stresses these bacteria or triggers these bacteria to produce their toxins, they can produce both membrane damaging toxins, right? So we'll just call those MD toxins. And they have the ability to produce superantigens. So by producing both of these at the same time, a patient who has TSS will go into rapid decline very quickly. Yeah. Yes, strep O and strep S desirine, that is correct. So these membrane damaging toxins start to break down host cells, and at the same time, you've got a super antigen being produced by the same organism that's causing an overstimulation of the cytotoxic response in T cells. So you've got all kinds of craziness going on in a patient at once, and this is going on in the circulatory system, so it's happening system-wide. So toxic shock syndrome was actually discovered back in the mid to late 80s, and it was discovered because of tampons. So there was an outbreak of women entering into hospitals with these sepsis-type infections, and it was always women, and they were developing strep and staph infections, and they were becoming systemic. And it was always, they finally kind of through interviews, they realized that these women were all coming in within a week or so after having their periods. And what was finally it led back to was the use of super absorbent tampons. So if a woman uses tampons, these were tampons designed by manufacturers for longer use, more than just a few hours. They were intended so a woman could have a tampon and use it for 12 hours. That's what they were designed for. Now we probably are like, ew, that's so gross, but that's what these tampons were designed to do. And the problem with that is that part of the body at that time of the month for women is very susceptible, and it's not sterile. Tampons, once you open the package, are no longer sterile, and normal flora such as strep and staph that would be found on those tampons were overgrowing in that amount of time. It was basically a big cotton incubator for these bacteria, and it was causing systemic infections later on. So it was originally discovered in tampons. That's why if you open a box of tampons, there's going to be warnings all over the place about TSS. That was where it was first discovered. Now we know that toxic shock syndrome can be caused by a multitude of different things, but that was a big outbreak, and it all had to do with the timing of the invention of these longer-term tampons, which are no longer on the market, by the way. Those are no longer found. It's a pleasant thought, right? Okay, so viruses. Now, viruses are interesting because they don't produce toxins, but they do have some activities that are really very interesting. One is viruses can avoid interferons. They do this by bringing with them, remember, viruses are just, a box of genetic material. So in their genome, they will actually have a gene for proteins that can neutralize interferon. So remember, when a virus infects a cell, our cells have those, they had the rig-like receptors, right? Yes, Wanda, they do not produce toxins. Not in the sense that bacteria do. So because remember, they don't metabolize, they don't consume, right? So a hemolysine, a membrane damage toxin like a hemolysine, is produced by bacteria in order to break down red blood cells and use them as a nutrient source. Viruses don't metabolize, they don't need a nutrient source. So they're producing proteins to just make more of themselves. They have evolved some genes to do things like neutralize interferons, but none of them are metabolic in terms of breaking something down in order to use it for energy, because viruses are non-living pathogens. Remember, these guys are not living. I know that in the news they're saying, oh, the virus is alive for a long time on surfaces. The virus is not living. A virus is viable. It may be viable, meaning it can still infect you. The viral spikes are still active or are still capable, intact, and can gain entry into your cells. So we'll talk about that in a minute. So these proteins, some of them can neutralize the interferon production. So when our cells get infected by a virus, remember that initiates this rig-like receptor to send a signal to the cell to start producing interferons. And so then in addition to their own genome, they'll have a gene in here somewhere like this that is encodes for a protein that'll attach to interferons and prevent the interferons from signaling the T cells and such, either memory cells, the macrophages, or recruiting other cells. Another, I wouldn't say popular, but another genetic evolutionary type of finding is some viruses can regulate cell death. Now remember, those rig-like receptors as well as nod-like receptors that we saw in innate immunity are found in the cytoplasm of all cells, right? So when a cell gets infected, these receptors here are going to get initiated, and they're going to tell the cell, hey, you need to produce antiviral proteins, and they're going to induce apoptosis. Those two receptors, when the viral versions of these receptors, the virus versions of these receptors are bound, they are going to initiate apoptosis in that cell. They're going to tell that cell, you are infected by virus, and instead of allowing you to become a viral factory, we are going to shut you down. We're going to burn this factory down to the ground so you cannot produce more viruses. That's what our immune system is doing, and that's what the cell is telling itself. The problem is that the virus may bring with it a gene for a protein that will shut off this protein here called P53. And P53 is the one protein that has to be activated in order to initiate apoptosis. So if P53 is not only not produced but neutralized, any that exists, then apoptosis will not occur. And it doesn't matter how much cell signal the cell receives from the outside and from the inside. If P53 is turned off or P53 is neutralized, apoptosis will not occur. It's the one protein that's very, very important for apoptosis to occur. When this happens, now our cell cycle, right, the cell cycle, M phase, S phase, G1, G2, those sorts of things, the cell cycle is now unregulated because the other job of P53 is to regulate cell cycle. It's a major component in allowing a cell to move forward in its cell cycle. And so without P53, we not only have apoptosis not occurring, but we also now have lost regulation of cell cycle. And this means that mitosis, DNA replication, all of that is now unchecked. So if the cell starts moving through the cell cycle too quickly and it's going through mitosis and DNA replication too quickly, then we start to see tumor formation, because we're going to start seeing mutations occurring, and the cell is no longer checking for those mutations. And so mutations will accumulate very quickly, and then we can start seeing cancer type formations. So viruses associated with p53 neutralization are very often considered oncogenic viruses. Some viruses can also neutralize the MHC class 1. Remember, all cells present antigen on an MHC class 1. So if they can neutralize the MHC class 1, they can prevent antigen presentation. So the cell may be giving off a signal, an antiviral signal, but the immune system can't recognize what that virus is because the MHC1 is not presenting it. So the cells will come over and be like, hey, what's going on? You're sending a signal. But the cell itself can't say anything. So they can't find the cell that is presenting that antigen. And if they can't find it, then that cell will just become a factory and continue producing viruses. And then last but not least, we have the avoidance of antibodies. Some viruses will actually use antibodies in order to gain entry into macrophages. We see that with bacteria as well. So they're inducing phagocytosis in order to gain entry into cells. And others, they can move directly from cell to cell. The measles virus, I think it's measles or mumps, actually the cells begin fusing together. Hang on, Desiree. So in like mumps, our cells will start fusing together, and that's how the virus induces this fusion, and so that's how the virus will move from one cell to the next by just starting to fuse the cells together. Desiree, class 2. Class 2 is not affected because class 2 is only found on antigen-presenting cells. So if the virus infects antigen-presenting cells, it could possibly be specific to an MHC2, but MHC2 is only found on macrophages, dendritic cells, and B cells. MHC1 is found on all cells, with the exception of red blood cells. Everybody good there? Okay. Does anybody have any questions? No? All right, so I want to do a bit of a class activity today. This is actually the end of the pathogenicity lecture. That's the last slide. It's not a particularly long lecture. Do you guys, before we go into a class activity, do you guys want to talk about any of this? Do you want to review any of it? Have any questions about anything on here at all? Yeah, Desiree.
Hello.
Hello.
Hi, hello, how are you?
I'm good, I'm good. What's your question?
I just had a little question. I tried to, I wasn't able to make it for Monday's lecture, unfortunately, so I tried to go back over it the following night and Tuesday, but I wasn't able to get into antibiotics. It keeps on saying to contact the mediator.
Huh, the antibiotic lecture? I'll have to take a look at it because it's on, it's actually a link from, I put it on YouTube, so it might be a YouTube issue. I'll go and take a look at it.
Okay, all right, then that was all. Yes, thank you.
Yeah, it says it was removed. Okay, Dominique, thanks. I'll take a look and see where it went. I might have made it private or something. I don't know. I'll have to go look. You know, you guys could have just texted me or emailed me. I'd have taken care of it.
So busy, I didn't want to
you know what? Honestly, it's easier if you send me an email because then I don't forget. Because otherwise I forget. So I'm going to write it down. I have a list every day that I write down because I am forgetting. See, yeah, all these due dates, I can't remember what class I opened them up on and which ones I didn't. Okay, I'll open the discussion I can do right away. Cochise postulates, Jalen. Okay, yep, I'll go over that. Let me find the, oops. I need to find the slide on it. Okay, Cochise postulates. Let me make my note here. I need to open discussion 5. Lecture. Not working. Okay, it's on my list for today. So when class is over, I'll go and take care of those two things. All right. Cochise postulates. Jalen was asking about this. In Cochise postulates, we want to make sure that that every time we have a disease, if we believe a disease is caused by a specific organism, I'm going to try to find a better picture for you. If it's caused by a specific organism, that we can prove that that organism is the cause of that particular disease. So let me see if I can find a better picture for you. I may even find a cool video on it. I don't want one that's long. Okay. Okay, none of those are any good. So I'm just going to find a picture and we'll do it together. I'm actually finding it for you. Okay. Come back here and I'll upload the pictures because I think this is something that might be a little easier to see with an actual image. So let me just upload that image for you. Okay, so I opened two of them. So let's start with this one. Hopefully it will render in a second. Okay, that one came out really blurry. Let's try the other one. Okay, so this is just kind of a cartoon version of Cochise postulates. And what's really going on here is first we look at a diseased organism, right? So we noticed that mice, and Koch was looking at this in Streptococcus pneumonia, and he was looking at this actually, I think, in tuberculosis in cattle. And he noticed cattle all had the same signs and symptoms. So what he did was he isolated bacteria from some cattle that were sick. And he looked at this bacteria under the microscope, and so he was able to not just identify it, but he described it. And he knew what it looked like and how it behaved and how it grew in media, those sorts of things. He then took some of that culture and he injected it into healthy cattle. Those healthy cattle then became infected with that bacteria, and they showed the exact same signs and symptoms, whether they died or they were just really sick, but they showed the exact same signs and symptoms. In those cattle that contracted the disease from actually being purposely introduced to it, he extracted samples from those cattle, and those cattle showed the exact same description of bacteria that he saw in the ones that naturally became sick. He then takes that bacteria and notices that, of course, it's the same signs and symptoms, and he can then say, hey, the bacteria that caused my original infection is the same bacteria that causes the infection in what we call a secondary host. So he was just trying to show that whenever you get infected by that bacteria, these are the signs and symptoms you're going to have, and this is the bacteria that's causing that disease. Where it becomes difficult and tricky is when we look at things like viruses and intracellular obligate bacteria, because and some other bacteria like syphilis. And the reason these are difficult to use Cochise postulates with is because they are not, we can't grow them in culture. They're hard to grow in culture. So right now it's a little easier with viruses. It's a little easier with viruses now because we have cell cultures. 1 second, Jessica, yes. We have cell cultures, and we can grow the bacteria in cell cultures, but that's, I mean, the viruses in cell culture, but that's not necessarily the same as actually using it or seeing it in an actual host. So to be more specific, and because viruses can be so closely related, they look at the genetics of the virus. So they would, instead of isolating a bacteria, they would isolate a virus from an organism, and then find other in the human population. It's not ethical to inject people, so we would find groups, right? Groups of people that are showing the same signs and symptoms. Those people, then we would take samples. And this is usually done in pathology first, so they would look at tissue samples. And of course, they oftentimes will figure out that it's a virus. And by looking at them in electron micrographs in tissue samples. They'll see that virus, and they will then start testing that virus using PCR. And they'll utilize primers in PCR. These are all concepts that we have covered throughout the semester. They'll use different primers for a whole host of different viruses. And so they'll throw in some retrovirus primers. They'll throw in, of course, coronaviruses. They'll throw in flu virus primers. They'll throw in primers for, I don't know, I'm trying to think of a papilloma type virus. And they'll do a whole bunch of tubes with primers from each of these different classes or families of viruses. And so let's take this COVID situation we're in. What China did was China ran PCR looking for retroviruses, and everything came up negative. So they said, okay, well, it's not a retrovirus. They ran it with flu viruses because some of the signs and symptoms are very confusing with flu, and it is flu season, but it kept coming up negative, so it can't be a flu virus. They ran PCR papillomavirus, and nothing came up in papillomavirus, but these signs and symptoms were also very similar to the SARS and MERS infections. So they ran PCR using primers that were common between SARS and MERS, that were common between the family of coronaviruses, and bingo, that's what they ended up with. They got positive reactions, and that's how they identified it as a member of the coronavirus family. But when they actually sequenced the entire genome, they saw that it was closely related to SARS and to MERS, but it was not 100% identical. So there are some minor genetic differences between them, and those genetic differences make a difference in its transmissibility in particular. So that's how they modified Cochise postulates. They use these basic concepts in Cochise postulates, but they apply it to a viral genome as opposed to growing bacteria in culture. This can also be done in toxin production, so they could isolate the gene for toxin production, knock that gene out, mutate it so it's no longer functional, and then see what happens when they grow the bacteria without that gene, and then they can say, hey, yeah, that's the bacteria that produces that gene, or that's the virus that has that particular gene. So it's that same kind of concept where they're comparing what they know already about a particular disease and comparing it to an induced version of that disease. Does that help, Jalen? Okay, great. All right, Jessica asked, could you compare that to the idea of isolating to identify bacteria? I'm not sure. You would definitely use the isolation procedure in this, right? So you would isolate the bacteria, but then you're going to grow the bacteria in the lab, and you're going to run multiple tests. So you look for metabolic properties of that organism, kind of like we're doing in lab right now, and then compare those metabolic properties to the metabolic properties of already known organisms, and then determine who it's the most alike. Nowadays, you would run PCR. So in PCR, let me pull up a PCR 96 well plate I want to show you something in PCR. Because sometimes if you just like see a picture of something, you're like, oh, now I get it. So I want to show you a picture. Come on, you can do it. There we go. Okay, this is what we call a 96 well PCR plate. Every single one, I can't draw when I share a picture like this, but every single one of those little holes, these little wells, you can see the little tubes, every single one of those tubes is a single sample. So let's say that we were scientists and we were doctors and trying to figure out this COVID thing, right? We were, in China trying to figure this out. They could literally run 96 different samples against however many, up to 96 different viruses. They could take a single patient sample and expose it to 96 different sets of primers and determine if any of those 96, within 3 to 4 hours, they could determine which of those 96 viruses their patient viral load had genes that in common with. So if I'm looking for COVID, let me download this picture and we'll, do you guys see what I'm talking about? No, Josu says no. Okay, so let me share the picture so I can draw on it. I think that would be better. Downloads. Okay, so here's the picture. Right? Everybody can see the picture now? Great. All right, so theoretically, I mean, they would do this in smaller numbers, but you can see each of these, right? We have, let me get my marker working. I have row A, row B, row C, row D, and then column 1, column 2. Think of a spreadsheet, right? Looks like a spreadsheet. And in PCR, do you remember PCR from biotech? PCR was when we were making copies of a specific region of a genome, and we were making tons and tons of copies of a specific gene in a genome. Well, remember, if somebody has a virus infection, I have my host cell that has its own genome, right? So I have my own genome over here inside my nucleus. But also, since I'm infected by a virus, I also have my viral genome. My virus is going to release its genome into the cell. So what I end up with, if I extract DNA, if this is a DNA virus, and I extract DNA from the cell, I'm going to get a combination in one of these tubes, in my patient sample. If I extract DNA from a patient sample, I'm going to get a combination of viral RNA or viral DNA as well as the patient DNA. So each of these tubes contains patient sample, right? So let's say, so we know that blue is patient, orange is flu, green is retro. These are virus families. This is, flu is called a flaviviridae. Purple will be, let's make purple papilloma. These are virus families. And then, of course, red will make coronaviruses. So each of my tubes here will contain patient sample. In addition to my patient sample, I'm going to add primers. So let's say primer-wise, I add retrovirus primers to this column here. So where I put the X is where the primers for this PCR product is. I put flu primers in this column. I put papilloma primers in this column. And I put corona primers in this column. So in this row, the red row, I'm looking for the presence of genes of genetic material that is only associated with coronavirus. In the purple row, I am looking for genetic information that is only found in papillomaviruses. In the orange row, I am looking only for genetic material that is associated with flu viruses. So only a flu virus would contain that gene sequence I'm looking for. And then of course, in the first row, the green row, I am looking for genetic material that is only associated with a retrovirus. I do this with several other viruses throughout the rest of the plate, right? So I could do all of these or all other different viruses I'm looking at as well. So I load this whole plate. I have 96 samples that I can use here. And I put this in my thermal cycler. I run all of the different temperatures and cycles to make tons and tons of copies of the genetic material that my primers are unique for. Remember, they're only going to be unique for a certain gene sequence or DNA sequence that would be found in that particular virus family. I'm looking at this on a computer, and then let's say I run electrophoresis. If I use electrophoresis, let me draw an electrophoresis box here. So if I run electrophoresis here, and I load each of my rows in here, I'm going to put my, let's see, I'm putting my retrovirus here. I'm going to put my flu virus samples here. I'm going to put my papillomavirus samples here. I'll put my coronavirus samples here. And then, of course, I need to run a control. So I put just plain patient, right? So maybe this row right here, I did a patient primer to make sure that I was positive for that my PCR was working at all. So if I run electrophoresis and my patient had a coronavirus, I will see no DNA bands in the retrovirus row. There'll be nothing there, 0. If I run the flu virus, I'm not going to see anything there either because my patient's sample doesn't contain flu virus. So there's no flu virus for the flu primers to detect, so nothing's going to happen. Same thing with the papillomavirus and, of course, the retrovirus. But in the coronavirus, I'm going to see bands here. And what I'm going to see is I'm going to see a single band that is telling me that the DNA that I am looking for is present in that patient sample. And I go back to my little map of my plate and I say row 4. What did I put in row 4? I put coronavirus. So if row 4 is the only place that I'm getting an actual sample from, then I know that coronavirus is present in my patient sample. My patient is infected with a coronavirus. Does everyone see how that works? So this kind of testing is now what they're doing. This is the confirmatory test for the coronavirus. So what they have done is they have sequenced the coronavirus genome, and they've actually come up with probably 2 to 3 different sets of primers so that what they'll see on that, let me erase this. I didn't mean to do the whole thing. I'm sorry, guys. Get my marker here and draw a new box. So in the PCR box now, what they're looking at is probably 3 different primers. 2 to 3 is usually what's done in diagnostics. I have not looked at the PCR primer, at the PCR test itself they're using in COVID. I'm not privy to that information. At least I can't find it yet. But they're looking at, so they'll find a band for this set of primers, they'll find a band for this set of primers, and maybe a band over here for this set. And so if they can find 3 different pieces of genetic information that are unique to a single virus, you can bet with pretty high confidence that that patient is infected with that specific virus. So let's, what I wanted to do is I wanted to break you guys out into groups, and I wanted you to actually go and find any information on COVID testing. So if you guys, I'll break you into groups, and I want you to kind of do a Google search together for about 10 minutes and see if you can't find a reliable source aside no CDC and no World Health Organization. I want you, I don't care if it's commercial, but it should be a fairly reliable resource to see if you can't find anything on COVID, like any research being done on new forms of COVID testing. I know they're talking about an LFD out there. They're finally talking about the antibody testing. See, we were weeks ahead of them. So on COVID testing, if you can't find anything on testing, you can also look for COVID treatment, because we are going to talk about treatments in our next lectures. So take a look at COVID testing or COVID treatment, not public health. I just want to know the science behind it. And what you'll do is look at it as a group and just kind of get a general idea, just a little like 1 or 2 minute introduction into what kind of test it is or what kind of treatment it is and how it works. Just a general idea. Are they using antibody? Are they testing for antibodies? Are they using PCR? Are they using genetics? What are they doing? And if you even can get into a little bit of the mechanics of it, that would be great. But I just want you to spend about maybe 10 minutes doing that in groups. Do you guys know how to share in groups? No. Okay, Adanya, let me show you guys how to share real quick. I'm going to break out here. I need to break into a new window to show you how to do it. So I'm going here. All right, so I'm going to share with you how when you guys get put into a group, as group members, you will be able to share either your screen, another Chrome tab, or an application on your computer. So I'm going to show you guys really quick how to do that so you can share information with each other. If you download a file, which is perfectly fine, you can do that. It will show up when we come back to the main room. So you can feel free to like take a screenshot if you want to, or download a file or something, and then we can add that. It has to be a PDF or a picture. So I usually recommend that you just take a screenshot. Okay, so I'm in a Blackboard Collaborate room right now. You guys can all see it, I hope. And once you're into the breakout groups, you'll have this little purple tab at the bottom right-hand corner. I want to make sure you can see that. Yes, okay. You'll click on this tab and that opens up your tools. Now, right now, you guys just have the chat room and the settings, but there's this share content that will show up when you get put into groups. So click on that and you'll click on share application screen. As long as you're in Chrome, I have to share a new screen with you now. As long as you're in Chrome, this little window is going to pop up. I'm going to show you what that window looks like. Come on, you can do it. Okay, so this little screen is going to pop up when you click that share application window. Now you can share your entire screen, which I don't suggest you do because it's going to get really confusing. Your best bet is to either open a browser in another window. You can share it as an application, like right now I'm sharing this lecture hall with you. Or if you have information opened up in another tab in Chrome, just select Chrome tab and it will show you all of the different tabs that you have open in Chrome, and you just click on it. Come on. It's in another window. You just click on it and then it'll share that tab in that window. So it's share application screen, and now you can see in my window here that we're looking at a tab that I'm sharing from another window. So you can share a tab, you can share an application, or you can share your entire screen if you want to. That's totally up to you. So you have that ability in your share rooms when you go to the share. Okay? Everybody good on how to share content? If you don't know when you're in there, you can just raise your hand. I'll see your hand raised and I can pop into your group and help you out. So if you have any issues, just raise your hand and I'll come and help you out. All right, so I'm gonna break you guys into groups now. I'm gonna do groups we have 4. Yeah, we have about 16 in here. So I'm gonna put you into groups of 4 and I want you to just work together for about 10 minutes and then we'll come back and one person from each group will just kind of speak real quick and tell me what they found in terms of either a treatment or a form of testing.